Clinical microbiology, quality assurance and quality control. So quality issues in clinical microbiology. Quality. If you think about quality, what does that mean to you? So if you have a quality product, it means it's probably a good product. And if the quality isn't good, then you're probably not going to continue to want to go buy that item. So quality has everything to do with whether a person or whether a customer is going to continue to come back and to continue to use a particular service or a particular item. Now that doesn't escape healthcare. So healthcare, everyone's got choices of where they want to go. And if we have a microbiology lab that's continuously misdiagnosing, then they're not going to want to return to that microbiology lab. They're not going to want to return to that healthcare facility, or their confidence in that healthcare facility is going to decline. So when we talk about quality, we're mostly referring to the validity of the results that are released from the lab. So whether the lab was able to diagnose the infection properly or not. So quality control are measures designed to ensure the medical reliability of laboratory data. So this evaluates the quality of the product, in this case, the lab data. So if we have a patient's specimen that comes down to the lab and we culture it on some media, but the media happened to be expired. And then we see that nothing actually grew. Well, if nothing grows, then we would report up to the floor and nothing grew. We would assume that, well, nothing grew, so maybe the patient doesn't have an infection. But there was a quality issue. And that quality issue was that the media that we used to culture the microorganisms was expired. So that's the quality issue, that's the problem, and that is where quality control would catch it. Quality control practices would have individuals recording what the expiration date was on the media, ensuring that all the media available for culture was not expired, and recording lot numbers of the media so that if a new lot number was tested, we want to make sure that that new lot number can actually grow what's intended to grow on it. So all of that is just related to one small aspect of quality control, but it's a very specific example. But there's lots of other areas in the lab, like using lab kits. We want to make sure that that lab kit is functional and that it works properly. So we use quality controller QC organisms that are known to produce a certain result to make sure that the lab kit is acting or behaving appropriately. Then we have a pre-analytical activity. Pre-analytical activity we're going to go into in more detail in the next lecture. But with pre-analytical activity, we're going to make sure that the specimens are labeled properly and identified properly. We're going to make sure that the specimen was transported in the appropriate amount of time, that the storage conditions were appropriate, that it didn't sit around for too long before we actually went to culture it. Analytical activity, that's where we start running like QC or known organisms alongside our unknowns to make sure that the kits or the media or the process is behaving as we expect. Post-analytical activity, that's double checking results, double confirming what we're seeing. If you're unsure of something, asking a second tech, hey, this looks like this to me. Does it look like that to you? Just double checking. Anytime that gram stains are reported out by someone that's newer, they always, in the lab, they always double check with another tech to make sure, I see gram-positive cocci. Is that what you see? And then double checking those results before those are released. Quality assurance is taking measures to ensure high-quality patient care. This evaluates the process. In other words, how efficiently the data is collected and processed. This doesn't really focus in too much on the detailed aspects of, is the kit expired or not? Is the kit functional or not? This is more big picture. How many steps did it take for the courier to deliver the specimen to the bench where it's going to be processed? when that happens. How many steps does it take to get to the analyzer? Or when we start culturing, how long does it take to actually report out those results? So quality assurance is kind of a much bigger umbrella of getting the results out accurately to the patient, but big procedural type viewpoint. While quality control are the steps, are the kits, are the personnel doing things accurately, and is everything look like it's going to produce valid results? So who are the customers in quality assurance? So the patients are customers because patients, like I said, are not going to return to a healthcare facility if they're continuously getting misdiagnosed. Physicians, if an ordering physician is getting incorrect order results or weird results or not getting results on time, then the physicians are the customers. Nurses. So the nurses are the ones that usually collect the specimens and send them down to the hospital lab, and then the hospital lab will evaluate the specimen and make sure that it's appropriate. If it was collected on the wrong swab or in the wrong transport container, or if the wrong test was ordered, that's something we would report back to the nurses and say, hey, this isn't okay. But on the opposite side of things, if a nurse collects it correctly and sends it down to the lab and we make a mistake and we drop it or misplace it or open it up and contaminate it, then we have to call the nurses for a recollect. And that's not always well received by the nursing staff. And then insurers. Insurance is only going to pay for you to run the test once. So if you have to run it twice, they may or may not pay for it. So insurance is another customer. Each have different perceptions of quality. So the patient wants relief from pain, the physician wants a fast turnaround time, the laboratory professionals want accuracy, and the insurers are looking for cost effectiveness. They're not going to pay for a $5 million molecular test when you can do it by hand with a rapid kit for $5. So measurable processes, you can read through these. You can go ahead and pause the video and take a look at these measurable processes. But the 3 stages of activities that affect laboratory testing: pre-analytic, analytic, and post-analytic. So I mentioned before, these 3 terms are going to haunt you through this entire program, and they're going to continue coming back when you get out in the profession. So these are really important. But pre-analytic would be like test ordering, order transcription, specimen collection, identification, and transport. Analytic is the actual testing of the specimen. And post-analytic, result delivery, result review, and the action taken on the basis of the result. Generally, guidelines associated with QC activities record all QC activities to prove their existence. So there are QC binders everywhere in the lab, and these QC binders require that all laboratory personnel check and report expiration dates on things, temperatures, room temperatures, storage temperatures. So fridges, freezers, make sure that all the temperatures are within the appropriate range. And it's not just that you're checking them, it's that you're recording them. So it makes the person physically write it down as evidence that not only were they checked, but they were written down and acknowledged. And then following that up with the person's name of who did it, when they did it, and making sure that it's done frequently enough so that there aren't large gaps of time where temperatures might be able to change. A list of tolerance limits to determine actions, and corrective actions must be recorded. So if a temperature is out of range, you say out of range, however, this was done to make sure that it goes back in range. So daily temperature checks are required on incubators, heating blocks, water baths, refrigerators, and freezers, and in the room in general. So a lot of our rapid kits now, and this maybe more so covered in like immunology and molecular diagnostics, but those kits require that the room be at a particular temperature. So if the room is too hot, the kit won't function properly. And vice versa, if it's too cold, the kit won't function properly. So having the room temperature checked is another one. Thermometer calibration. use the National Institute of Standards and Technology calibrated thermometers, and they're calibrated by batch on arrival to the laboratory. After successful calibration, the thermometer may be put to use. Repeat calibration should not be necessary. Already calibrated thermometers are available for purchase, but you have to have a certificate of calibration on file for each thermometer. This seems crazy, but this is something that we keep track of in the laboratory because temperature is so important for things operating and functioning properly. And we'll learn a lot more about that, like I said, when we talk about the molecular diagnostics and immunology courses, how important temperatures are when we're running certain rapid kits. Equipment QC, tested for proper performance at the appropriate time intervals. Does it work correctly? So if we have a molecular test for identification of a particular pathogen, we would run QC organisms, QC quality control organisms, which means that we know that they're supposed to either produce a positive result or a negative result. We would run those frequently. Sometimes it's daily QC, sometimes weekly. Very rarely would we run them only monthly, but daily or weekly, depending on how many of these tests we run in the lab. Preventive maintenance program must be established. Repair or replace parts before problems arise. So then here you can read through some of the frequency of equipment testing. Every lab is going to have a very particular protocol of how frequently equipment needs to be tested, but you can pause the video and take a look at each of these. And then when you do get a job offer and you become a medical lab tech, when you're on the job, you can see their own QC logs, and it will tell you right on the QC log how frequently it has to be done. And this is an example of a preventive maintenance log sheet. So, for example, check refrigerators and freezers for outdated material. So in January, it was done on this date, February this date, March this date, and so forth. Media QC. So if you recall from just general microbiology, media is the plates that we grow bacteria on. Anytime that we get a new batch, it must be tested to document performance and sterility. So we incubate a plate, and if anything grows, so we incubate a plate that has nothing on it, that we didn't put any bacteria on it, and if we see growth, we know it's been contaminated. We also want to test certain organisms on certain types of selective and differential media to make sure that the selective and differential media is performing correctly. Records are maintained for 2 years, and this is mandated by the Clinical Laboratory and Standards Institute, so CLSI. Required to have a statement of QC from the manufacturer for all media that the laboratory will not retest during use. So complex or specialized media may require further QC testing, like chocolate agar, Neisseria medias, Campylobacter media. Other factors that are recorded are the moisture, sterility, breakage, and appearance of the media. Retested commercial media, you can go ahead and pause the video and kind of look through here. This is if a laboratory mandates that the media must be retested. Complex or in-house made media, testing with organisms expected to grow or produce a reaction. We also test the pH and sterility, and QC organisms should be the most fastidious. So you want to get the toughest to grow, and if it can grow on the media, then we know that the media is good. Testing of media should be different for primary plating media than biochemical or subculture media. So primary culture, dilute organisms, subculture, undiluted. And you want to establish the expiration dates. So here's sample media observation log. So for this one up here, BAP, abbreviated blood agar plate or sheep blood agar. They're recording the lot number, and then they're checking moisture, sterility, appearance, and breakage. Reagent QC, so reagents tested each day of use, positive and negative controls. So if something is supposed to turn blue in the presence of a particular organism, you're going to use a QC organism which is known to turn blue. You're also going to use a QC organism that's known to not turn blue, and make sure that the one that's supposed to be blue turns blue and the one that's not doesn't. And you have to repeat that every day that you're going to use that particular reagent. Some examples include stains, bacitracin, catalase, coagulase, oxidase. Oxidase is the one that I was kind of thinking of when I was given the blue example. So if we have an oxidase positive organism, it should turn blue. You use the oxidase reagent. If it doesn't turn blue, you know you got a problem with the oxidase reagent. So workstation QC. So this is just a sample like blood culture workstation quality control. So gas pack jar, the heat catalyst, change desiccant weekly. And then you can see the heating blocks, the temperatures that were recorded. And as long as the temperatures are maintained within a particular range, you're good to go. And antimicrobial susceptibility QC. So susceptibility testing is done on ATCC control organisms, American type culture collection. Some variables exist, so the antibiotic potency, agar depth, evaporation, cation content, pH, instrument failure, or inoculum concentration. So those variables can be controlled for a little bit. Antibiotic potency, so most of the time we're going to get pre-made antibiotic discs with a known concentration of antibiotic, or we're going to get pre-made plates that have known antibiotic concentration wells in them. The agar depth, this can be controlled by buying manufactured agar instead of by making the agar yourself. And instrument failure, if we see that the instrument has failed, we would have to repeat the test. We'll talk a lot more about antibiotic susceptibility testing later on in our lecture series here. Susceptibility testing, control organisms conducted daily for 20 to 30 days. After this, only once a week. Minimum inhibitory concentration values must be within 1 log dilution of the expected MIC based on the CLSI guidelines. Keep QC results as long as the test is in use and at least 2 years after discontinuation of the agent. So here's some recommended control organisms for susceptibility testing. Personal competency, so competency assessment techniques, direct observation, the ability of an individual to perform a task accurately and effectively, review of worksheets and work cards, and written examinations. So when you leave school, you go get a job offer and you've been working in the clinical lab, you routinely have to get retested and reevaluated for your ability to perform the tests accurately. Proficiency testing is a good way of doing that. So they will give you an organism and it's an unknown, and they will watch you work up that organism to produce results. And then you have to identify that organism correctly using the correct procedures by the end of this. And you will have all the resources that you typically have. So if you use protocols at work, you're going to have those protocols in front of you. This is not done by memory. You get all of the same resources that you would normally have. It's just you don't get the resource of another person. So you have to work this up on your own, given the resources and workflow charts and all the things that you would normally have working up a normal patient. So all tests performed must be subject to PT twice per year. PT samples should be processed, handled, and analyzed the same as patient samples. Another form of PT QC is review of all work by another laboratory scientist before finalizing, and this can catch errors before releasing results. Competency is verified and documented. You have recertification each year. Continuing education is also required. And then this is just competency documentation, a quick sample of what it might look like. Stock cultures, so stock culture sources, commercial sources, patient isolates, proficiency testing isolates, ATCC, bacterial strains. The culture should be grown in broth first, and then the aliquots are stored for 1 year in the freezer. That's to prevent mutations from subculture. Broth should not contain sugars. The bread or bead method, store beads in vials containing 1 ml of thioglycolate broth and store at -70. All rules and procedures must be available in a QC manual. The QC manual must be at each workstation. Review it at least once a year and revisions as needed by a supervisor.